Hi, welcome to MTHFR support. Today we will be going over the AGT M235T gene and how it interacts with MTHFR. Medical disclaimer. You understand that any information is found within our website is for general and educational and informational purposes only. You understand that such information is not intended nor otherwise implied to be medical advice. The AGT gene encodes angiotensin, a secreted glycoprotein produced mainly by the liver and belonging to the non-inhibitory serpin family. It is not a classical enzyme. Instead, it is a substrate for renin and the renin-angiotensin system, or AS. Renin cleaves the N-terminus of angiotensin to generate angiotensin 1, which is then converted by ACE to angiotensin 2 and other downstream peptides that regulate vascular tone, sodium balance, and blood pressure. Because angiotensinogen sits at the top of this cascade, its concentration is effectively rate limiting. Small changes in AGT levels can shift angiotensin 2 generation and downstream vascular and inflammatory tone. M235T is strongly linked to higher circulated angiotensinogen levels and increased RAS activity. Multiple studies associate the T235T allele and especially the TT genotype with higher risk of hypertension and coronary artery disease, like via increased AGT expression and angiotensin 2 driven vascular and inflammatory signaling. In that sense, regulation here is about substrate availability and gene expression, not about cofactors or turnover at an active site. Angiotensinogen itself doesn't require a vitamin or mineral cofactor to function. The key regulatory levers are gene transcription, hormonal control, for example, estrogens and glucocorticoids, and renin availability. MTHFR lives in a different biochemical neighborhood, one carbon metabolism, but they converge at the level of vascular biology. MTHFR helps convert 5,10-methylene tetrahydrofolate to 5-MTHF, which is needed to remethylate homocysteine to methionine. Variants that reduce MTHFR activity can raise homocysteine and alter methylation capacity, which has been linked to endothelial dysfunction, oxidative stress, and higher cardiovascular risks in some contexts. When you put an AGT M235T background tending toward higher RAS tone and vascular constriction, together with a reduced MTHFR efficiency tending towards higher homocysteine and impaired endothelial resilience, you're layering two different axes of cardiovascular stress, hemodynamic load and biochemical oxidative load. The interaction is not a simple if A and B, then C, but a probabilistic stacking of risk pathways. RAS activation, inflammation, endothelial function, and redox balance all intersect. AGT M235T can push the system towards higher angiotensin 2, more vasoconstriction, and more inflammatory cytokine signaling. MTHFR variants can push the system towards higher homocysteine and altered methylation of genes involved in vascular tone. antioxidant defenses, and coagulation. In a person with both, the threshold for hypertension, microvascular damage, or brain and kidney vulnerability may be lower, especially if lifestyle, diet, or other genes like GSTs, NOS variants are also nudging the system in the same direction. Because of that complexity, peer-reviewed research is essential but not sufficient on its own. AGT literature will often focus on blood pressure, coronary artery disease, and RSAS modulation. The MTHFR literature will focus on homocysteine, folate status, and methylation. Each paper tends to isolate one variable at a time in one population under one set of assumptions. Real people don't live in those controlled conditions. They bring in diet medications like ACE inhibitors, ARBs, diuretics, PPIs, metformin, etc. Comorbidities, stress, sleep, and other polymorphisms that can amplify or buffer these effects. That's exactly why working with a practitioner is so important. A good clinician isn't just treating AGT or treating MTHFR, they're reading the pattern, blood pressure trends, inflammatory markers, homocysteine, kidney function, lipids, symptoms, family history, and your lived experience. They can decide when AGT M235T finding is clinically meaningful. In some with early, difficult to control hypertension or strong family history of cardiovascular disease, for example. And when it's just background noise, they can also prevent well-intentioned but risky self-experimentation, like aggressive methyl donor supplementation in someone with high blood pressure and no monitoring, or ignoring RS-driven risks because I'm just working on my methylation. In practice, the togetherness of AGT, M235T, and MTHFR is less about a single pathway diagram and more about how your vascular system, redox balance, and blood pressure regulation respond to life over time. Genetics sets the baseline, environment and choices modulate the expression, and a practitioner helps you interpret the whole picture, anchored in peer-reviewed evidence, but tailored to your specific physiology rather than a one-size-fits-all protocol. Come and visit us at mthfrsupport.org. MTHFRsupport.org can be a valuable starting point for individuals and practitioners looking for community-driven information, educational materials, and potential guidance on navigating MTHFR and related genetic variations, including AGT M235T. For anyone exploring this complex topic, it is always a good practice to combine information from advocacy and support organizations with peer-reviewed research and personalized care from a qualified healthcare practitioner. The interaction between genetics, biochemistry, and supplementation is highly individualized. We hope to see you over at mthfrsupport.org. Have a beautiful day or evening wherever you are.